Okay, so in the next 1 hour, we are going to discuss what all the questions you have attended in your today's test and this test which we are conducted in our Google form, right? So, and results will be declared after this session. So, it's time for you to evaluate yourself. What are the mistakes you done in your test paper? So, you had totally 50 questions. So, we are going to take exactly next 1 hour. We are going to rush through all those 50 questions. So, all these 50 questions are interrelated to each other. When you are able to attend this 50 questions, that means that you are ready for attending any question that is coming from your muscle physiology. Unfortunately, today we couldn't conduct the nerve physiology. In the coming week, maybe on Monday or Tuesday, we will conduct the nerve physiology test also. To begin with, first question, all are related to the negative lucitrophy except. To answer this question, first you need to understand what is lucitrophy. Lucitrophy means relaxation after a contraction. In which muscle? So we tell inotropy, lucitrophy, chronotropy, all these things are going to be discussing about the cardiac muscles. So inotropic, that means that the force of contraction, chronotrophy, all these are going to be discussing about the cardiac muscle. Similarly, we are discussing the relaxation phase of a cardiac muscle. Unlike your other muscles, like your skeletal muscle, your cardiac muscle is not going to be following the same phenomena. That is, when a nerve gets excited, acetylcholine comes. This acetylcholine is going to be binding with the ligand-gated sodium channel. That is going to be again increasing. Then your sodium channels, voltage-gated sodium channel opens. Then your ryanodine receptor then first your dihydropyridine receptor, then your ryanodine receptor. This is how your entire chain goes. That is what we discussed in the lecture itself. So same concept I'm going to apply here, but the thing is, instead of sodium channels, instead of uptake of sodium, then calcium channel getting opened, here the calcium itself stimulates the calcium channel inside your sarcoplasmic reticulum. That means, unlike your sodium channels, here your calcium channels are the one, the calcium are the one that stimulates your dihydropyridine receptor. From dihydropyridine receptor, again your mechanical ryanodine receptor opens. So here, what is the difference? The dihydropyridine receptor does not stimulate by the sodium ion, but here it is going to be stimulated by the calcium ion. Number one, you need to understand between the difference between the cardiac muscles and your skeletal muscle. Number two, when the calcium is released from the sarcoplasmic reticulum, this is going to be regulated by calstabin protein. This can be asked as one MCQ in future. If you are going to get, this can be asked as a MCQ. Calstabin. They inhibit the ryanodine receptor and they decrease the calcium ion release from your sarcoplasmic reticulum. Now, you need in order to have a relaxation, you all know that like skeletal muscle, you have the sarcoplasmic endoplasmic reticulum, calcium, Sarca, ATPase, isn't it? So that Sarca, in order to act, okay, that is going to be regulated by the phospholumban. So phospholumban is a molecule. What it does is it inhibits your Sarca during the contraction phase. When there is a relaxation, this Sarca, okay, wanted to get activated. So in order to Sarca getting activated, the inhibiting factor has to be removed, right? What is the inhibiting factor here? Phospholumban is going to be the inhibiting factor for Sarca. This phospholumban should be phosphorylated so that it releases your Sarca, so that Sarca can do the job of reuptaking your calcium and thus facilitating the relaxation of your cardiac muscle. In order to phosphorylate your phospholumban, you need norepinephrine. Norepinephrine is the one that is going to phosphorylate your phospholumban. Thus, it releases the Sarca. Once Sarca is released, that is going to facilitate the relaxation of your cardiac muscle. Okay, that is going to be promoting the lucitrophy. So, lucitrophy means relaxation. Now, who is promoting lucitrophy here? Norepinephrine is promoting lucitrophy. Phospholumban, what it does? It is going to be negative lucitrophy. So, what are the negative lucitrophy factor? Phospholumban is going to be a negative lucitrophy factor. Okay, now consider if this Sarca is not working, then there is no reuptake. Then it becomes negative lucitrophy factor. Correct? Lucitrophy and relaxation inhibit எப்ப ஆகும்? இந்த Sarca work ஆகலைன்னா உங்களுக்கு inhibit ஆகும். Okay, number one. So negative, when there is going to be mutation in your circa and dysfunction of your circa, that is going to lead to improper functioning of circa, lead to negative lucitrophy. Similarly, your calcium is removed not just by your circa mechanism, but it is also removed by sodium calcium exchanger. When there is a defective calcium removal by the sodium calcium exchanger, that will also lead to defective relaxation. So that will cause negative lucitrophy. Okay. Similarly, when there is excessive calcium present within the cell, if at all, let's consider, if at all there is more calcium that is present within the cell, then also what happens is there is more excitation. Until the calcium is going to be there inside the cell, that is going to cause excitation. Therefore, relaxation phase varadu. Appa, first moon option paranga. Number one, excess of calcium within the cell. It contributes to negative lucitrophy, that is contraction. Negative lucitrophy is opposite to relaxation, that is contraction. Similarly, defective removal of calcium through sodium calcium exchanges, that is again going to cause contraction. That means it is a negative lucitrophy. Similarly, third one, improper functioning of circa, again it is going to cause negative lucitrophy. Excess of catecholamine, what it does, we know that norepinephrine going to phosphorylate your phospholamban. Phospholamban is an inhibiting factor of your circa. When this phospholamban is removed, circa is going to get activated, reuptake of calcium. Therefore, there is going to be relaxation. So the answer is, what is the answer? The answer is excess catecholamine. The answer is excess catecholamine. So everyone clear, right? What is the answer? The answer is excess catecholamine. Now, moving on to, that's the second question. Protein connecting Z line to M line. Okay, they have given four points here. They have given kinin, desmin, actin, titin. Okay, now what we will do is we will see one by one. First one, kinin they have given. What is a kinin, pa? Someone, can you tell me in the chat box, what is kinin molecule? Okay, then I will ask the second thing, what is desmin molecule? Actin molecule na enna? Idu nalathu inge nama paathutomna, next if they are going to ask a question, we will be able to answer. Remember, kinin is a polypeptide, okay, which you are going to see in the entirely different system. So kinin is not going to be playing any role in the skeletal muscle contraction. Kinin is going to be playing an important role. They are going to be present in the plasma. This kinin, what it does, okay, this kinin are going to be like a kallikreinin. Okay, this is going to cause bradykinin formation. You all know kinin beta 2 receptor. This is the where you will be having a bradykinin formation. So kinin receptors act as na pharmacological activity nadako. So that is entirely is going to go for the ACE production. Okay, so bradykinin on the side it is going to go. So kinin is not playing any role in your muscle contraction. We will leave that now. Number one. Number two, what was the question given? Desmin. What is a desmin? It is the intermediate filament. Okay, desmin is the first one, kadayadu. Second one, intermediate filament. Okay, what it does is it connects Z line to the plasma membrane. It connects Z line to plasma membrane. So when the question asked, okay, which is going to connect the Z line to plasma membrane, you should be able to answer. It is going to be your desmin. What is actin? Actin is going to be nothing but your thin filament. Okay, actin is going to be nothing but your thin filament. That is actin. So this actin is going to be your I band. Actin is going to be your I band. That is going to be present where? Actin is going to be present as a I band. That is going to be present over the Z line. It is going to be present over the Z line. Appa actin no kadayadu. Finally, you are going to have titin. What is a titin? A titin is a muscle spring, we call it as, responsible for elasticity, which connects Z to M. Which is going to connect Z to M. So answer is titin. So titin, what are the other points you need to remember is, titin is also called as muscle spring because it is responsible for the elasticity of the muscle. It connects your Z line to the M line. Thus, it provides a scaffolding for the sarcomere. Thus, it is going to providing scaffolding for the sarcomere. Is it clear? So you have completed the second question. Next, moving on towards the third question. Myosin actin filaments are taken in place by who is going to take your myosin actin? Okay, anything? What is the other role of your titin? Anyone knows? I think I told you this. Myosin and actin are going to be taken in a place. It means that your actin myosin both are going to be molded together by this one protein, very important structural protein, that is your titin again. Titin is a structural protein, you need to remember. Remember that tropomyosin, troponin, these two are regulatory protein. They are going to be regulatory protein. Okay, regulatory protein. And your actinin, it binds your actin to your Z line. So actin to Z line bind பண்றது யாருன்னா இந்த actinin. அதே titin என்ன பண்ணோம்னா they connect your Z line to your M line. இப்போ Z M கனெக்ட் பண்ணதுன்னா நடுவுல யார் இருக்காங்கன்னா thick filament, thin filament. That is going to be your actin and your myosin. Therefore, இந்த actin myosin ரெண்டு பேரையும் ஒரு இடத்துல உட்கார வைக்கிறது. So it's going to bring both of them together. Who is going to bring both of them together? Your titin. So the answer is titin. Okay. Next, sarcomere extends between. So what is the sarcomere? So you know that this is going to be your myofibril. Okay, this is going to be your myofibril. Okay, this is going to be one Z line. You know that this is going to be one Z line. This is going to be one Z line. Between the two Z line is your sarcomere. So the answer is sarcomere extends between two Z line. It's very clear. So what is A band? What is I band? Someone, this is going to be just a revision. A band I band A band which is made up of mainly myosin. A band is the one which is made up of mainly myosin. So you know that this is going to be a M line. There's going to be a Z line. M line mela, you're going to have like this. Z line mela, you're going to have like this. So there is one particular place where there is going to be a mingling of your two cells. So and the where you're going to have just your myosin alone. That is called as your A band. Okay, then what is your I band? I band means actin filament alone, actin filament alone. So they are going to be like this place alone is called as your actin. The one which is where only your actin filaments are there. They are going to be I band. So sarcomere extends between two Z line and not A band or I band. Okay, next question. In a muscle contraction, all are true except. I again told you this very important one. So this particular point, I was always repeating you that this question will be asked in your test. I told you, right? So in a muscle contraction, all are going to be true except. So while you read about the muscle contraction, you need to always remember four important points. Okay, at the end of the contraction, A band will always remain same. A band will always remain same. Okay, remains unchanged. So this option is correct. H zone will be disappearing. H zone will be disappearing. So that is going to be the next one. Why H zone disappears by anyone? What is H zone first of all? First tell me what is H zone. The region where A band, where only thick filament will be present. But during the excitation contraction, what happens is the entire thin filament comes into your, it comes very close to your M line. Therefore, this H zone is going to be disappearing. So that option is also correct. Third one, I band becomes widened. Now tell me, okay. So this is going to be your M line. Okay, this is going to be your Z line. These two are going to be your Z line. Now what happens is, you told that there is going to be a thick filament like this. There is going to be a thick filament like this. You also know that there is going to be a thin filament that are going to be present in between like this. Okay, you're going to have a thin filament like this. Similarly, this side also. Okay, just consider. So it's going to be like this. Now what we are meaning to tell is, okay, now what we are meaning to tell is, what is the A zone? A band this area. This area is called as your A band. Okay, this is going to be called as your A band. Similarly, your I band. Okay, your I band is going to be the area where only actin is going to be there, or only thin filament is going to be there. Okay, that is going to be your I band. Okay, A band is going to be one, your thick A band and another region spanning from one thick filament to other thick filament. So A one thick to other thick. Okay, you all know that there is no change in the thick filament. Only your thin filament overrides your thick filament. Therefore, there is not going to be any change in the A band. H zone disappears. That is the area where exclusively you're going to have thick filament. That exclusion is not going to be there because of the your thin filament are going to enter into the thick filament area. It's going to override that area. Therefore, H zone will be disappearing. Your I band is this area. Your I band is this area. Now tell me, your I band is the area where there is a region between the external end of A band till your Z line. External end of A band. A band is the one thick filament to other thick filament. So external end of the A band ku Z line ku irukra and the gap da on the I band. இந்த ஐ பேண்ட் என்ன ஆகும்னா இந்த ஆக்டின் மட்டும் இருக்கக்கூடிய இடம் வந்து it will get shorter. Instead இங்க widerன்னு போட்டிருக்காங்கல்ல, it's not going to get wider. Instead it is going to get shorter. So it's going to be getting shorter. So that is what your mistake is. Similarly, whenever there is going to be a excitation contraction, your sarcomere is going to be shortened. Okay, what is your sarcomere? Extending from one Z line to other Z line. When your sarcomere shortens, two Z line comes closer to each other. Therefore, the answer for this question is, in muscle contraction, all are true except I band becomes wider. That is the answer. I band becomes wider. That is going to be your answer. The site where myosin yet binds to the actin and skeletal muscle, where you all know that. Can you all just think of it? Can you all just think of it? So what happens is, you're going to have an actin. Okay, so I have already, I think I have drawn it and showed you. So this is going to be your actin thick filament. Similarly, you will be having like this. Okay, something like this. It is going to be your actin, this is going to be your thick filament. So this thick filament is usually, they are going to be covered by the tropomyosin. It is going to be covered by the tropomyosin. Okay, the site where the myosin head binds to the actin. The site where the myosin head is going to cover to the actin. At which side they are going to be covering to the actin? Someone? Again? Can someone tell me? Yeah. Can you try? Can you try in the chat box? Question ketangana, we will try to rule out. Troponin. Does troponin have any role in the actin? Yes, it is there. How it does? So when the calcium is going to come and sit on the troponin I. Okay, so you have this troponin I, troponin C, troponin I, you have different things, right? How this entire process occurs. So whenever there is going to be a muscle contraction, for a muscle contraction take place, so calcium comes out, it combines with the troponin C, activates your troponin T activates your tropomyosin. Then this tropomyosin moves. Okay, tropomyosin moves, and that is the place where your actin myosin interaction takes place. Now, myosin yet binds to the actin and skeletal muscle at what place? They கேட்டிருக்காங்க. அதுக்கு வந்து troponin is not the answer. Calcium is not the answer. Now, choosing the best answer, tropomyosin is the one which is going to cover. At the place of tropomyosin covering only, your myosin is going to bind with the actin. Therefore, the right answer is tropomyosin. Now, actin's active site is covered by. This is very direct question. Tropomyosin is the one that is going to be covering your actin's active site. Okay. Now, thin filament consists of all except, always remember, your thin filament is going to be made up of three important structures. Okay. What are the three important things? Number one, actin. Number two, tropomyosin. Number three, troponin. These are the three things which I told you in the lecture. So this question is going to be removed. This option is going to be removed. Now you are left with two important options. Myosin, desmin. Which is the answer? Very easy. The myosin is going to be your thick filament. It's not thin filament. Okay, desmin, what is that? You know that desmin is an intermediate filament that is going to be again sitting on your thin filament only. Therefore, your answer is going to be your myosin. Okay, so myosin is a thick filament. Okay, now troponin C mediated function is of the which of the following. Now remember, you have trop C, you have trop I, you have trop T. Okay, you have three important troponins in your skeletal muscle. Now they have asked you what is the troponin C mediated function. Okay, so remember troponin C troponin T venga act பண்ண போகுது அப்படிங்கறத கேக்குறாங்க. Remember, I'll tell you one easy thing to remember. Troponin C is for calcium. Troponin C is for calcium. Okay, troponin I is going to be on actin. Okay, troponin T is going to be on tropomyosin. If my question was trop C, trop T, I can choose tropomyosin. But now it is not going to be trop T because it's the trop C is given. Therefore, tropomyosin option is ruled out. And everything is going to be on the actin. Nothing is going to be on the myosin. So myosin is ruled out. The question itself gives you one idiotic thing is the question gives you option as troponin. This option is actually wrong. Troponin is actually, it cannot be there. So the best answer for this question is going to be your actin. So the best option for this is going to be your actin is going to be the right answer. So binding of calcium with troponin C causes a conformational change by removing trop I and trop T and exposes your myosin binding site. That is going to be your actin. That is going to be your actin. Okay, next. Calcium ion in skeletal muscles are. Very good question. So calcium ions in your skeletal muscles are going to be terminal system. What is that? I'll tell you. So you have a sarcotubular system in your muscle, namely T tubule, and you're going to have a L tubule. Remember, you're going to have T tubule, you're going to have L tubule. The T tubule are going to be present in the sarcolemma, that is a plasma membrane of your muscle, and L tubule are going to be present on the sarcoplasmic reticulum. So what happens is this is going to be a T tubule. So that is going to be invaginating inside your sarcolemma, that is going to be your cytoplasm of your skeletal muscle. Okay, so this is how your motor end plate is going to be there. Okay, and this is how it's going to be there. Okay, now here you're going to have a sarcoplasmic reticulum. So this is going to be your sarcoplasmic reticulum that is going to be present between the two T tubule. So the calcium ion, okay, sarcotubular structure. So this is going to be a T tubule. And where is your L tubule? L tubule are going to be present in the sarcoplasmic reticulum. Okay, which is going to store and release the calcium. Okay, wow. So இந்த மாதிரி ஸ்டோர் அண்ட் ரிலீசிங் கால்சியம் இருக்கு பத்தியா, அதுக்கு பேர் வந்து டெர்மினல் சிஸ்டர்ன்னு சொல்றோம். So it's called as terminal cisterns. So the L tubules are the sarcoplasmic reticulum which stores and releases the calcium. That L tubule is called as a terminal cistern, and from this terminal cistern is only calcium ion in skeletal muscles are released. Therefore, calcium ions in the terminal muscle is going to be terminal cisterns. Okay, this is going to be the answer. Now, calcium is pumped back into the sarcoplasmic reticulum by, you know that sodium pump, it is going to pump your calcium outside. It's going to pump the calcium from your sarcoplasmic reticulum into your cell. But here it is what we are telling it, calcium is pumped back into sarcoplasmic reticulum, so sodium pump is not there. You also know that ryanodine receptor is also going to be the mechanical receptor that pumps outside. It's going to pump out the calcium from the cell, so ryanodine receptor is also not there. Sodium pump is also not there. So SERCA, that is sarcoplasmic endoplasmic reticulum, you're going to calcium ATPase. So that is going to be a SERCA. So it's going to be very important. They are going to be playing an important role in pumping back your calcium into your sarcoplasmic reticulum. So the answer is your SERCA. Okay. Next, when skeletal muscle contraction ends. When is your skeletal muscle contraction will be ending? When your ion move out of cytoplasm. No. When the ions are going to move into your cytoplasm, into your sarcoplasmic reticulum. So that is number one. Acetylcholine absorbed from the NMJ. You know that when acetylcholine is going to be absorbed by at the neuromuscular junction, that is going to induce the ligand-gated sodium channels. So from your ligand-gated sodium channel, it is going to induce your voltage-gated sodium channel. It's going to induce a voltage-gated sodium channel. On inducing a voltage-gated sodium channel, there is going to be an induction of your dihydropyridine receptors. That is going to again induce your ryanodine receptor. Ryanodine receptor that is going to release the calcium. That is going to release your calcium and going to cause contraction. This is the entire chain. So skeletal muscle contraction doesn't end when the acetylcholine is absorbed. So that is also not the answer. Closer and indrawing of receptor. I'll have this as one option. The fourth one is going to decrease the calcium outside your reticulum. Very right. So this can be a good answer. Decrease the calcium outside the reticulum. When there is going to be decrease the calcium outside your sarcoplasmic reticulum, automatically the entire skeletal excitation process is going to be stopping, because calcium is necessary for activating TROP-C. TROP-C activates TROP-I. TROP-I activates your TROP-T, and TROP-T removes your tropomyosin. Tropomyosin covering remove ஆனாதான் உங்களுக்கு skeletal muscle contraction நடக்கும். இங்க அது நடக்காது. அதனால this can be option. But now my question is, why closer and indrawing of receptor is not the answer? Remember, there cannot be indrawing of receptor and all. Okay, and when there is going to be closer receptors present, that is going to be meaning that there is more number of receptors going to be present. When more number of receptors present, more number of acetylcholine comes in to take place. So this is not, if at all they have given the same thing as circa, closer circa, or if they have given indrawing of what receptors, particularly if they have given, I would have chosen this option. But the best answer is going to be your decrease in the calcium outside your reticulum. Now, which of the following triggers muscle contraction? So, which of the following going to trigger your muscle contraction? I have already told you, calcium binds with the TROP-C. Okay, TROP-C order தான் bind பண்ணும். Okay, TROP-T binds with your tropomyosin. TROP-T order binds with tropomyosin. TROP-I binds with actin. Okay, so and ATP breakdown does not cause, ATP breakdown is also going to be one, but ATP breakdown is not going to cause muscle contraction. TROP-C order calcium, so this is ruled out. This is ruled out. You need ATP. I accept for ATP is required for the process of your muscle contraction. Calcium binding troponin C is also required. But which is the best answer? Which triggers your muscle contraction? If you look into calcium binding trop-C is going to be the best answer. Therefore, I am choosing that. All of the following are true about your excitation contraction coupling, except. Okay, let's discuss, right? Number one, acetylcholine is released at the nerve terminal. This is one question. Is it right? Acetylcholine is going to be released at nerve terminal. This is 100% right. So this option is right. Calcium is pumped into the sarcoplasmic reticulum during relaxation. Yes, of course, this is going to be pumped by circa. This is going to be pumped by circa. Therefore, the answer is true. Therefore, the answer is going to be true. I am having no doubt in that. Okay. Third one. The calcium is released from the sarcoplasmic reticulum during contraction. Of course, yes. Calcium is released from your sarcoplasmic reticulum during contraction. Fourth option was calcium binds with the T-tropomyosin to initiate muscle contraction. No. Your calcium is going to be binding to TROP-C. TROP-C goes and binds to your... So that is what I am telling. This one simple concept, they have asked us numerous questions in your exam. TROP-C is going to go and activate your TROP-T. This TROP-T is going to activate tropomyosin. Tropomyosin moves from its place, which facilitates the actin-myosin interaction. Actin-myosin interaction. So now you know the answer. It is going to be your calcium binds T-tropomyosin to initiate the muscle contraction. Okay. Next. This is the wrong one. Okay, this is the wrong answer. So that is why they have asked except. Always see this except and then answer. Myosin filament has a fixed length. What is a myosin filament? What is the length of the myosin filament? Okay, remember myosin is going to be always 1.6 micron. So that is the right answer. 1.6 micron. That is the right answer. Next one, ATPase activity is present in. Okay, ATPase activity is going to be myosin head contains actin binding site, and they are the catalytic site that hydrolyzes the ATP to ADP and your. So this ATPase is going to act on the myosin. They are not going to act on the actin. They are not going to act on the troponin. They are going to act on the myosin. So myosin lenna. So this is going to be your this is going to be your head. So in the head ATP enna panna, it hydrolyzes to ADP and phosphate. This phosphate is the one that pushes your head of your myosin back, and that is going to cause contraction. Okay, number one. And then most common type of calcium channel of skeletal muscle. Remember, most common type of calcium channel that is present in the skeletal muscle in a human is going to be your L type. So remember that. Next question. Okay, duration of maximum contraction depends upon. Duration of maximum contraction depends upon relative refractory period. I'll tell you what is relative, what is absolute. Absolute, not no stimulus, no matter how strong it is, it will not get excited the muscle during the absolute refractory period. So at this point, there is no contraction. So let's see here. So I'll just take it. There is going to be a slowly contraction is going to take place. Then there is going to be relaxation is going to be there. At this relaxation pace, okay, while this is going to get relaxed, until this finally gets relaxed and attained to minus 70 millivolt. And the minus 70 millivolt திரும்பி resting membrane potential வர்ற வரைக்கும், how strong you give some stimulus, it is not going to be doing any action. So in the period, the absolute refractory period. Relative refractory period na, okay, stronger than the normal stimulus, supra-threshold stimulus, it sometimes produces action potential. So therefore, contraction is possible. Therefore, in a relative refractory period, maximum contraction is going to be depending upon the relative refractory period. So, and the threshold, so minus 55 millivolt varra varaikum, you call it as the absolute refractory period. From minus 55 to minus 70 millivolt irukla, and the period relative refractory period. And the time la, in case innor stimulus kuttengana, the resting potential pogamiye adutha stimulus aarambichidum. So it's like a summation. You have read about the summation, right? So that is the answer. So answer is going to be relative refractory period. Twitch of a single motor unit is called as fasciculation. Remember, fasciculation is the contraction of individual motor unit. Fibrillation, it's a contraction of individual muscle fiber. Okay, next one. Fine irregular contraction of individual muscle fibers. I told you already, when there is going to be a contraction of your muscle fibers, you call it as fibrillation. When there's a contraction of individual motor unit, that is fasciculation. Contraction of individual fibers, you call it as fibrillation. The force of muscle contraction can be increased by all of the following except. By increase the frequency of activation of motor neuron, of course, yes, it can. It is called as summation. You can increase the force of contraction. Okay, increasing the number of motor units getting activated. Yes, again, increasing the number of motor units, again, it is going to get summated and going to cause increase in the force of muscle contraction. Increasing the amplitude of muscle action potential. Remember, action potential obeys all or none phenomena. Once threshold level is crossed, whatever action potential is generated, they are going to be having a constant amplitude. So increasing the amplitude of action potential doesn't have any role. Doesn't have any role. Okay, and recruiting larger motor fibers, again, it is going to cause increase in the frequency of activation and increasing the number of motor unit. Therefore, there is going to be increase in the force of contraction. Active tension in muscle depends upon. Active tension of muscle is going to be depending upon what? it is going to depend upon the number of muscle fibers A, or whether it's going to be depending upon by the number of motor units or aerobic capacity or length of the muscle fiber. Always remember, active tension of the muscle depends upon the number of motor units recruited. Number of motor units recruited. So that is the answer. Tetanic contraction is due to the accumulation of calcium. Exactly. So when the skeletal muscle is stimulated, before its contraction gets over, it responds to another contraction. This is called as summation. Why there is a summation? Because of accumulation of calcium ions. Such type of summated contraction is called as complete tetanus. I think I told you yesterday. Post-tetanic potentiation is due to what? Post-tetanic potentiation. So why there is a potentiation? Because of the availability of calcium. Again, same concept. When there is accumulation of calcium ion, it is going to summate without getting relaxation. So answer is going to be increased availability of calcium. When the tension in the muscle fiber is maximum, its length is called as, again I told you this, optimum length. Remember, so few important terminology you need to understand. What is active tension? If muscle is stimulated, actin myosin going to interact, muscle contracts and it develops active tension. What is passive tension? It will be elastic protein titin is there. That is going to cause without stimulation, there is going to be some tension. That is called as passive tension. So what is the total tension? Sum of active tension and passive tension. What is resting length? The length of the muscle at which active tension is maximum. That is called as resting length. It corresponds to the segment of BC in the graph. Equilibrium length அப்படினா என்னன்னா ரிலாக்ஸா இருக்கும்போது ஒரு லென்த் இருக்குல்ல, because of this titin, that is called as equilibrium length. Okay, இங்க கேட்டிருக்கிறது tension in muscle fiber is maximum is going to be there at the optimum length. Now, white fibers are present in where? Remember, the motor unit where innervates very few muscle fibers in the muscle concerned with fine, precise, graded movement. Okay, where all you will be having a fine, precise, graded movement? Extraocular muscle. Okay, in your hand muscle. So these are the places where you need a very motor unit muscle fiber concised with a fine, precise, graded movement. It is going to be present in the extraocular muscle and hand. So white fibers are present in the extraocular muscle. So you know that white fibers are called as fast fibers. White fibers are called as fast fibers. Less mitochondria are seen in. You know that less mitochondria, again it is going to be seen in the white muscle fibers. So okay, white muscle fibers, red muscle fibers have many mitochondria, white muscle fibers have less mitochondria. All are true about red muscle fiber, except again red muscle fiber will have more mitochondria, right? It has more myoglobin, it has more oxidative capacity. Okay, but the red muscle is going to be mainly depending upon the red muscle is going to mainly depending upon the aerobic metabolism. It is going to mainly depending upon the aerobic metabolism, no anaerobic metabolism. Therefore, glycolytic metabolism nadakadu. Okay, glycolysis nadakadu. Okay, so that is why B option is wrong. Next, type 1 muscle fiber or type 1 muscle fiber or red muscle fiber. Again, some same related question. How many muscle fiber are there in one motor unit? Remember, in a one motor unit, okay, in each motor unit, you are going to have 3 to 6 muscle fibers in the eye. In eye, there is going to be 3 to 6 muscle fibers in the extraocular muscle. So 3 to 6 related answer is going to be your 5 for this question. So this question is again asked in your okay, everything is going to be again a previous year question only. Whatever we are discussing here, 2014 they have asked this question. Fast fatigable motor unit recruited first, last, during relaxation, none. Remember, the answer is last. That is called as a size principle. So smaller the diameter, recruited will be first. You know that fast fibers, okay, so slow fibers are having a small diameter, they will be recruited first. Fast fibers are going to be having a bigger size. So a bigger size will be recruited at the last. Therefore, the answer is going to be last. Which of the following is not sarcolemmal protein? You know dystrophin, you know dystroglycan, sarcoglycan. இது எல்லாமே வந்து பாத்தீங்கன்னா dystroglycan, sarcoglycan, even I can call it as merosin is going to be one of them. Dystrophin is going to be one of them. All these are going to be in that dystrophin. All are going to be part of dystrophin glycoprotein complex. Dystrophin glycoprotein complex. So, okay, so in the complex, in the perclan, it is going to be a extracellular matrix glycoprotein. It's a extracellular matrix proteoglycan or glycoprotein. This is not part of your sarcolemmal protein. So a spontaneous release of acetylcholine at the neuromuscular junction produces what? So when the acetylcholine is going to be released, it is going to cause motor end plate potential or miniature motor end plate potential. It does not stimulate action potential. It's going to be just a local potential. So post-tetanic potential kadayadu. It is produced by calcium. Resting membrane potential kadayadu. Again, that is also mistake, taken wrong. Okay, next one. End plate potential following which law? End plate potential is actually following depolarization. End plate potential are local potentials. They are always depolarizing. Once they cross your threshold, action potential is fired. Action potential is the one that follows all or none phenomena. Okay, hyperpolarization. So propagation. So this is just a depolarization. That is the right answer. So latent period of muscle twitch is 10 meters a second. So this is very important. Contraction period is 40 millisecond. Relaxation time is 50 millisecond. What is the tetanizing frequency? Remember, tetanizing frequency is equal to 1 by contraction period. Contraction period. Remember, your contraction period is just 40 milliseconds. So TF is equal to 1 by 40, which is equal to 25 hertz. So in the question is so in the point இது எல்லாமே waste. உனக்கு தெரிய வேண்டியது contraction period என்ன 40 milliseconds. So 1 by contraction period is your frequency of your tetanizing frequency. That is 25 hertz. That is going to be your answer. Next, human cardiac muscle is going to be contained. Always remember, your cardiac fiber is going to be very different that of your myosin heavy fiber. Your muscle, cardiac muscle is going to be containing alpha and beta isoforms of your myosin heavy chain. It's going to consisting of alpha and beta MHC. Okay, so both are going to be present in atria. Alpha isoform, but it is going to be predominating. Alpha is going to be predominating in atria and beta is going to be predominating in your ventricles. But both are going to be present in your cardiac muscle. So both alpha and beta heavy chains of myosins are going to be present in your human cardiac muscle. Next, heart muscle with true or false. This is the question. Remember, your heart muscle acts as a syncytium. Yes, of course. Okay, heart muscle is going to acting as a syncytium only, and it is going to have a gap junction. Yes, and they are going to be branching. Yes, but remember, your heart muscle or your cardiac muscle are going to have only nucleus, or you're going to call it as a single nucleus. Therefore, this question, this answer, this option is wrong. Therefore, heart muscle true all except is the answer. So this is going to be the correct answer. What is the true about cardiac muscle? Remember, your cardiac muscle is going to be having gap junction. So gap junction are present. So this is going to be wrong. Remember, so you also know that it is going to have a uninucleus. So it's going to be having a large nucleus, central nucleus. It is not spindle shaped. It's again wrong. They are not arranged in sheets. Okay, remember, so your cardiac muscles are actually not arranged in sheets. Your cardiac muscles are going to be, they are not arranged as a sheet. They are going to be a different pattern. Okay, they are going to be arranged in a very different pattern. They are arranged as a intercalated disc. So they are going to be arranged as a intercalated disc. Okay, it's going to be arranged as an intercalated disc. So this is wrong. This is wrong. And what is the shape? So it is going to be the shape of your cardiac muscle is going to be again like a disc. It is going to be not spindle shaped. It is going to be disc shaped. And that is also wrong. So it has a large central nucleus. That is your right answer. So what is the function of phospholamban? Phospholamban inhibits your SERCA. This was the first question which we saw. Is there anything? So it regulates the calcium entry into sarcoplasmic reticulum. Yes, correct. Is the 39 right? 39D right answer? Yes, of course. So it is going to regulate your calcium. I think I have told you about this already. Okay. Now, regulation of smooth muscle tone is persistently influenced by, remember, it is persistently influenced by latch bridge mechanism. I told you your smooth muscle can be maintained in a prolonged state of partial contraction with very little use of ATP because of your latch mechanism. So where your myosin crotch bridge remains attached to your actin for some time after cytoplasmic calcium concentration falls. This phenomena is very, very important in case of your vascular smooth muscles like in your intestine, etc. Now, smooth muscle cell membrane is again it is going to be called a sarcolemma. As skeletal muscle cell membrane smooth muscle cell membrane எல்லாருக்குமே நம்ம sarcolemma தான் பேர் வைக்கிறோம். Okay, most distinguishing feature between your skeletal muscle and your smooth muscle. Remember, troponin. There is no troponin in your smooth muscle. You are going to have a calmodulin. Your smooth muscle contains tropomyosin, actin, myosin, everything, but troponin is not going to be there. Which one of the following acts to increase the release of calcium from endoplasmic reticulum? Remember, from the calcium to release from endoplasmic reticulum, I think smooth muscle cells also, I think you would have read, isn't it? Okay, for when there is going to be binding of acetylcholine receptor to muscarinic receptor, there is going to be influx of calcium. Influx of calcium leads to activation of calmodulin dependent myosin light chain. When there is a calmodulin dependent myosin light chain, that is going to cause phosphorylation of myosin, that is going to cause actin myosin interaction. For this actin myosin to get interacted, okay, so there is going to be for getting interacted to, for this calcium has to get released, isn't it? And the calmodulin activator இருக்குல்ல, அது எது மூலமா ஆக்ட் பண்ணுதுன்னு பார்த்தோம்னா ionositol triphosphate மூலமா தான் ஆக்ட் பண்ணுது. So ionositol triphosphate is the one that helps in the mechanism that helps in releasing your calcium from your endoplasmic reticulum. Increase in the cytosolic calcium from intracellular storage during smooth muscle contraction is due to, remember, always calcium அப்படினு எடுத்தாலே they are going to be IP3DAG mechanism. Always calcium நீங்க எடுத்தீங்கன்னா IP3DAG mechanism தான். கண்ண மூடிட்டு நீங்க இதுக்கு டிக் பண்ணலாம் ஆன்சர். Calmodulin, what it does, it is going to activate your protein kinase. So calmodulin activates your protein kinase. So that is your calmodulin dependent myosin light chain kinase. This leads to phosphorylation of your myosin and actin myosin interaction takes place. Okay, calmodulin acts by what it does, phosphorylation of your myosin by protein kinase. So next one, what is the function of calmodulin? That is your CAM. CAM is nothing but your calmodulin activating protein. So CAM is going to be activated by, again it is going to be activated by the activation of protein kinase, activation of your protein kinase. So activation of your protein kinase, isn't it? So true regarding the excitation contraction coupling in your smooth muscle. Remember, presence of cellular calcium is essential for muscle contraction. So cells kulla muscle irukano. Troponin thevaiya thevailla. Calmodulin, that is there. Sustained contraction occurs at higher concentration of calcium. Phosphorylation of actin is required for contraction. No, not required. Presence of cellular calcium is required. Yes. Next, smooth muscle physiology different from skeletal. How? Anyone? Troponin is going to be absent. That is the answer. The nerve impulse which leads to initiation of smooth muscle contraction. Another, your nerve impulse which leads to initiation of smooth muscle contraction cause opening of your calcium channel, and that leads to increase in the calcium concentration. Calcium concentration increases, automatically it's going to cause depolarization. That is what I told you. So causes opening of your calcium channel which leads to increase in the calcium, and that is going to cause contraction. So these are the 50 questions that we have seen in the test, and the answers of these 15 questions are briefly discussed. When you can understand and answer these 50 questions, I'm very sure that you will be able to write your nerve muscle physiology well. Okay, the nerve physiology, again, I will discuss nerve physiology in this weekday. I will announce in the group and I will discuss those questions also. We will have a test, and formally we will be discussing like what were we done for the muscle physiology. Okay, thank you so much. If you have any doubt, post it in the chat box or you can post in the group. So the entire concept of this learning itself, I am also learning few things from teaching you all, and you're also learning something. So this is a two-way interaction. Just give me your feedbacks. Just make sure they just give your suggestion about the questions. So next time I will be improving and give you a good type of questions. And this is ultimately going to help you for the proposed next exam, which is going to be taking place for your final years of the present year 2019 batch. So of course you will be writing next only. So we will make sure that we will all give a good try in all MCQs from next time. So this time participation was less. I think next time I want all the 1,000 who are all present in this group should actively participate in the test, so that it is benefiting you, it is benefiting everyone. Okay, thank you so much. Have a good day.